Hello, my name is Dr. David Hayes. I'm from Eastern Kentucky University in the Department of Biological Sciences. Today I'm here to talk to you about some of our lab's work on eDNA metabarcoding in Mammoth Cave, and specifically we were looking for the Kentucky cave shrimp. So first off, I'd like to acknowledge some folks. First and foremost, most of everything I'm going to talk about today in terms of our Mammoth Cave Kentucky cave shrimp project is from the master's thesis of Andrew Stump. Yeah, this is all his. He did all the lab work and all the analysis, and so he deserves all the credit. He is currently employed at Kentucky Division of Fish and Wildlife Resources. A former student of ours, Grayson Patton, who's now an instructor at Surrey Community College, also helped us out a lot with some bioinformatics. Carrie Allison at U.S. Fish and Wildlife Service, who's pictured here, helped us out a lot with getting funding, as well as getting our hands on some little legs of these shrimp. The National Park Service has been great, specifically Rick Olson and Rick Toomey. The University of Kentucky's Advanced Genomic Technologies Center was also a great help at the time. And then we've also had a variety of other undergraduates and graduate students that helped Andrew in the field or helped us out in the lab. So environmental DNA, this idea has been around much longer than this paper, but this is the first paper that was able to detect the DNA of macrobiota, so a large organism. So I mentioned this stuff has been around for a while, mainly in the realm of bacteria and fungi, but in 2008, Faisatola et al. Was able to show that they were able to detect bullfrog DNA from water samples. And since then, there's essentially been an explosion in the field. And it's hard to keep up. I used to be really good about keeping up on papers that were coming out on eDNA and a variety of things, and they are coming out too fast and too quick. So I definitely can't say I've read every single eDNA paper like I could back in 2010. So environmental DNA is a source. It's not really a method, it's a source. It's a biological substance that can be recovered from an environmental sample. So it's DNA that can come from, say, soil, sediment, ice, feces, water. Oftentimes it is degraded, basically due to UV light, bacterial degradation, stuff like that. It will break down, and as soon as it's released from the organism, it begins that process of breaking down. So metabarcoding is a technique, and it is sort of derivative of the field of DNA barcoding, where you take certain genes that are taxonomically informative and use them for identifying things to the species level. Metabarcoding is similar, except that you're taking that sample rather than from an individual, you're taking it from a mixed sample. And so oftentimes you're using a short taxonomically informative marker. And so this allows you to sequence lots of taxonomically informative markers from, say, like a jar full of insects, or from a soil sample, or from a water filter. So you can look at multiple different types of organisms at one time. And so it extends the utility of these molecular markers to community level samples. And traditionally for animals, we use the cytochrome oxidase 1 or CO1 gene, although for other groups, there's been various other barcodes that have been useful. All right, so eDNA studies generally come in two flavors. First up is single species screens, and initially this used endpoint gel electrophoresis. You use species-specific primers, and if you've got a band, you had a detection. Nowadays, it's more commonly used with quantitative or real-time PCR, where you have species-specific probes. So with that, you're basically getting presence or absence of that one species. You can also get copy number of, the number of copies of DNA that you've got in that sample. However, there's much debate as to whether or not that information is informative. Then we have multi-taxa screens that use metabarcoding that I talked about initially. You generally use broader primer sets, and those primer sets might be for, say, a family, or maybe they work for all invertebrates, or maybe they work for fish. There's no sort of one universal, all-encompassing primer set. It's really for particular subsets of groups of taxa. And then generally you're going to use next generation sequencing. For the most part, almost all of the studies that do this use Illumina sequencing. We haven't really seen other sequencing technologies like Oxford Nanopore's MinION and PacBio. We haven't really seen that being used with environmental DNA samples yet, and it's likely because those technologies are really for long read sequencing. And eDNA, we're generally looking for short fragments. So Illumina sequencing is sort of ideal for environmental DNA samples. So here's a general workflow I like from a publication, Bick et al. In 2012. Here is your community of interest. It might be in the substrate here, it might be in the water. So you go and collect that sample. I'll talk about some collection methods in the next couple of slides. You'll extract that DNA. I'll also talk about some methods for extracting DNA. And then you'll amplify a taxonomically informative marker. Now, what constitutes a taxonomically informative marker might depend on your question. So if you're looking for really broad community stuff, you might pick something that's highly conserved and would work across multiple phyla. If you're looking for species level resolution, you might pick a primer set that is more tailored towards the species level resolution for your particular taxonomic group. So there's not just one particular marker or one particular primer set. And you might have to design a new primer set for the level that you're interested in. Once you amplify that, then you can do some high throughput sequencing, and you can do community analysis, similar to other ecological community analysis. I won't really get into that today. All right, so since this field of eDNA emerged in 2008, it has really blown up, and it's been applied to a whole lot of different organisms. Hellbenders and carp seem to be very popular for eDNA studies, but they've been variously used in lots of other groups, all the way up to whale sharks you see here. And so it's only been maybe in the last 5 years or so that we start to see its use in karst-dwelling organisms, so cave and groundwater-dwelling organisms. But it is showing to be useful in these organisms that are particularly hard to survey. And that's a sort of a key thing with environmental DNA. You want to use it for things that are difficult to survey for. You're not going to use it for things that are really easy to survey for or wouldn't take you much time to find. So karst environments are sort of like the ideal habitat to apply environmental DNA surveys because they're difficult to get to, they're difficult to sample. All right, so that brings us to our study, Mammoth Cave, shown here in green. This is the Mammoth Cave National Park, as well as the Western Kentucky University's nature preserve here in the Mammoth Cave system. The Green River sort of cuts through this karst system. And as many of you know, Mammoth Cave is the largest cave system, largest known cave system in the world. So this project was basically brought to me by U.S. Fish and Wildlife Service. They were interested in seeing if they could apply environmental DNA to search for Paleomonias scanteri, or the Kentucky cave shrimp, or sometimes called the Kentucky blind cave shrimp. As you can see, it has no eyes and lacks pigment. It is a stygobitic cave obligate, and it is only one of 3 extant members of the family Atiidae in North America. It's been federally listed as endangered since 1980, and it is endemic to the karst watersheds in and around Mammoth Cave Park. So these basins here, these are subterranean basins where shrimp have historically been found. So they've gotten at least one record from each of these 10 basins. Now, It might occur in other basins in this area, but they haven't turned up yet. And I'll talk about that a little bit later in some future work. So our main goal was to see if we could detect it primarily based in places where it's already been found, at least for a preliminary shot at this. So we sampled 11 sites between 2012 and 2013. Seems like forever long ago. We still haven't gotten this published, but Andrew's thesis is available under Google Scholar. So you can at least find that to reference that if you're interested in some of the more details of the sampling here. So we filtered 40 liters of water using 20 0.7 micrometer glass fiber filters. There's a variety of filtering substrates out there. Since this time, various reviews have been published. Some of them, there might actually be better substrates to filter through. And so we basically did 2 liters through 20 filters, 20 individual filters. We then folded those filters up and preserved them directly in the cell lysis buffer of the kit that we had decided to use. So there are a variety of ways to filter your samples. This is the method that we came up with. This is this blue part here is a filter head. This costs about 35, 40 bucks. And then it's connected to a hose to this hand pump. This is a gas transfer pump, liquid transfer pump, oil transfer pump that you can get at pretty much any auto parts store. I like designing pieces of field equipment that can easily be replaced if you're in the field. This filter head, not so much, but this pump, very easy to go find another one if it fails on you. So in this filter head, this is what will hold your glass fiber filter. So basically you stick this head in the water, and this is your outflow that will allow you to measure the volume of water that you pump through. This is that filter head. We've taken that, we've filtered our 2 liters through it, we fold it up so that all the sort of brown stuff is on the inside and protected, and then we'd add our cell lysis buffer to that. This is us sampling. This is Owl Cave, and I believe this is Echo River. You can sort of see that hand pump there. Occasionally we would also use this Liquivac. I really like this thing because it has the reservoir contained right in there. This is made for draining the oil out of your lawnmower, but it works great, except for the fact this thing isn't really made to handle water. So over time, the valves and the rubber stuff dry out, and basically the pump will fail. For the most part, my students seem to like this, the liquid transfer pump seems to get a better flow rate. This is just us, again, in some of the surface springs. So not everywhere we sampled was cave environment, some of them surface springs where occasionally shrimp have popped up in these places. So this is the method we use. One of the most common questions that I get from people when they want to do eDNA stuff is, how do I collect the water samples? There's not any one way. You really want to pick it based on your system. And so there are some people that do the mechanical filtering like we're doing here. Some people go with something as small as these little syringe filters. This might be ideal if you're sampling small little ponds or wetlands. I would stay away from these hand pump vacuum filter pumps. They break, they're cheap, they're not really great. Even if you get sort of a heavier duty one, doing this in the field is a lot of work. If you want to, you can also, again, rig up that Liquivac like we did. If you want to go power tools route, you can rig up a peristaltic pump head to a cordless drill and take that in the field. And that's pretty efficient as well. Same basic principle. You'd have a filter on one end, you'd have an outflow for measuring how much that volume that you filtered. And if you really want to go hardcore, there's a company that is partnered with Smith Root for making a backpack eDNA sampler. This would not be something I would want to carry into the depths of Mammoth Cave, but if you're doing a lot of surface sites and you're doing lots of eDNA, as your job or constant monitoring, then this might be an appropriate tool. For us, we were able to pack all this up into 1 gallon Ziploc bag that we were able to sterilize and have lots of repeats or lots of duplicates of that. So we weren't sterilizing equipment in the field in between sites. And then if you don't want to filter in the field, you can always haul water. And for some projects, we decide that hauling water is the better option, particularly in really turbid sites where you don't want to be sitting on the bank of the Mississippi River filtering by hand. And there are other methods for handling non-filtered water. There are extraction methods. You can centrifuge and pellet and flocculate and other types of methods. We won't really get into all of them there in here, but they all have their benefits in different systems. All right, then DNA extraction. We use the Mobio Power Water Kit. This kit has since proven to be less ideal for eukaryotic eDNA. People that do microbiome stuff really like the Power Water Kit. It's since been purchased by Qiagen, so now it's the eDNA Power Water Kit. But some studies have shown that this kit results in reduced yield and reduced detections for eukaryotic macrobiota. So it could be that this bead beating step, this bead bashing, it might be that it shears eukaryotic cells a little too much, or at least shears the DNA, compared to bacteria, which are much tougher and requires a stronger lysis. So it might be good for bacteria, but not great for eukaryotes. It worked out okay for us, we think. We got results. And so there are a variety of other methods. My lab has used traditional organic extraction, like not quite phenylchloroform, but similar. We've used CTAB chloroform extraction, basically liquid-liquid extraction. We've also used spin columns from a variety of manufacturers. We haven't quite used magnetic bead capture. There are some, it seems that magnetic bead capture doesn't work out as good as some of the other ones, but a lot of them haven't been tested explicitly. So you're really going to be picking the extraction method that you use best, I guess. And so there are a variety of studies comparing different methods, and some find better yields than others. And I've basically used them all. And sometimes I just end up making a judgment call as which one we're going to do for this project. All right, primers. So a little reminder, there's no such thing as a perfect primer set. And so oftentimes they're going to be taxon or group specific. Some of them that have been published that say they have broad applicability, we find that they do have certain biases. They might amplify, say, one order of organisms better than others, which might mean the data set that you would like to be evenly representative across taxa in your system actually shows more of one thing than are actually there because of that PCR bias. And some simply don't work reliably in the lab. There's some well-known published primer sets that I simply cannot get to work in the lab. And oftentimes you may need to design new primer sets for your particular target organism. And to do that, you need reference sequences. And so sometimes that is a major hurdle, which was an initial hurdle in this project in that there were no published sequences for Paleomonias canteri. And so we had to wait until U.S. Fish and Wildlife Service got us a tiny little leg, actually 2 tiny little legs. You could barely see them in the tube. They were collected under permits and given to us. And so we had this precious tiny amount that we could then generate our own reference sequences for. But you need those to be able to develop primers. All right, so an example workflow here. This is from Vasco Elbrecht. I would highly encourage you to check out his publications. They're mostly doing what I call bugs in a blender type work, where you take a sample like a macroinvertebrate sample or a malaise trap sample, and you'll homogenize those insects and then do your process after that. But the process is really the same if your sample is an environmental DNA or leaf litter sampling. But basically you'll filter your DNA, or however you're collecting your sample, and then homogenize it. And sometimes you have to homogenize that filter. And then you often want to do replication. And so you might end up deciding to say, you got your filter, you might cut it into quarters and say, we're going to extract 3 of them and then save 1 of them. Or for our sites, we did 20 separate filters. That took us about 2.5 hours per site for filtering. And so we were able to replicate that way. You might also replicate after that in a number of PCRs, but replication is really important here. But a lot of times it's going to come up to time and budgeting constraints. So then you'll do your DNA extraction using any variety of methods. You will likely want to quantify, see how much DNA that you have in there. And then you'll do your initial, you'll do a 2-step PCR. Here's PCR 1 and PCR 2. I'll show that on the next slide. But basically this allows you to first amplify your target region, the marker that you're interested in, and then 2, do your library prep. And library prep basically means you're attaching the things to the sequences that allow you to, or that you're attaching to your PCR product that allow you to run it on the sequencer itself. There's a couple of different ways that you can do this. The cheapest and probably most straightforward way is doing this 2-step PCR program. There are commercial kits, but they are more expensive. Then a lot of this stuff is stuff that I make the sequencing company do. So I'll get it up to this point, I'll do that second amplification, and I say, you take over from here and send me the data. So they'll do some quantification and pooling. And that pooling, because this fusion primers here, they actually contain unique identifiers in there. So you can basically identify a sample later on bioinformatically. So basically I can insert a little snippet of DNA and say, this goes with site 1, this one goes with site 2, this one goes with site 3, do my amplifications, and then I can pull them all into the same tube essentially, and then sequence them. And then later on, I can come back in the bioinformatics and say, all right, separate out site 1, site 2, site 3, or sample 1, sample 2, sample 3, things like that. So you're able to pull a lot of specimens on a single run, or a lot of samples on a single run. Variety of cleanup and then bioinformatic processes. So this is that two-step PCR protocol. So you have your locus-specific sequence. So in our study, this might be something for CO1 invertebrates with this overhang sequence. This is an adapter, and this will just hang out there. It is non-complementary to your target strand, but it will get incorporated in there. You might have to do a little cleanup here, and then you're going to do a second PCR reaction with a new set of primers that is complementary to that old overhang. And in this one, you are actually incorporating the index. This is a sample identifier. So this might be for a site or a single sample. And then the adapters here indicated by P5 and P7, these are what's going to actually bind onto the flow cell of the sequencer. And again, there are some other methods to do this library prep. This is the cheapest though. All right, so eDNA sequence identification, generally they fall under these 3 types. First, the simplest blast, many of you are familiar with just doing DNA barcoding. You get a genetic sequence, you run it through this blast algorithm, and it searches all of GenBank and sends you a list of the most closely related things, or the most genetically similar things that were found in GenBank. There's also operational taxonomic unit clustering, I'll talk about in a second, and phylogenetic placement. So OTU clustering is a way of reducing the size of your data set, which is critical with next-gen sequencing techniques. So basically what it's saying is, I want to, your sort of sequences are in a cloud, and I want to cluster them based on similarity. So I'm going to do these operational taxonomic units. I'm going to say, let's put everything in a group that is within at least 3% similar to one another. All right, so each member in this identical OTU is within 97% similar to each other. All right, in some cases, you might have OTUs that sort of have some overlap. So you might not be able to distinguish that species from this taxonomic unit to that taxonomic unit. So anyway, the way this reduces your data set is you create these little clusters, and then you just pluck one out and use that to identify. So essentially you're using that one sequence as a proxy for the identifier for all those other sequences in there. And this is really useful for reducing the size of your data set. Now, whether or not you lost some, informative things in there, we don't really know. You'd have to go back through and do some more digging in there. But that's there's a cost and a benefit to each of these analyses. And that's one of the costs there. But the benefit is, again, you reduce the size of your data set. You pluck that one that's either the consensus or the centroid, and you identify that one. Then we have phylogenetic placement, and all three of these can be done in combination, and we in my lab have found reasons to do all three of these things at various times. With phylogenetic placement, you basically can build a guide tree from references. So let's say, for example, I've done this with freshwater mussels. I'd make a phylogeny of freshwater mussels, and then these are my sequences that I've pulled out of water samples, and then I want to place them on the tree to figure out where they cluster. And this is kind of nice because it doesn't rely on a somewhat arbitrary cutoff line of 3%, like in operational taxonomic unit clustering. But again, none of these are sort of better than another. It's really what question you're trying to address, and you're going to pick the appropriate tool for that. So just general things that are available for bioinformatics. If you take a next generation sequencing workshop, or you're probably going to be introduced to CHIME, possibly Mother. CHIME seems to be a bit more popular these days. So these are tools that were developed for microbial ecology, and they're nice packages in that they have basically all the software and programs to do your cleanup and then your identification. Galaxy is a free cloud-based software program. This is great for sort of visually mapping out your workflow and your data analysis. Sometimes it has problems with the server going down, and of course it will do that while you're in class with students trying to complete an assignment. More recently, mBrave has come along, and this is really nice. You basically upload your raw sequence data. You can set some parameters. It is a little black box. They give you a little bit of leeway in some of those parameters, but then basically it relies on the BOLD database, Barcode of Life database, and will basically do analysis for you and show you sort of what matched those databases on BOLD, or those things on BOLD. There are a variety of other software programs as well. If you're into R, there are a variety of packages for doing this on R, assuming you have the computational resources to do that on your machine. Same thing for all of these. Computational resources is a huge thing with this, depending on how big and how big a data set you're working with and what your sequencing depth was. Reference databases for all these things are key. So you need them for your primer or probe design, and you need them to identify what those sequences are. If you do not have a reference sequence, then it's going to be very difficult, if not impossible, to identify the reads that you get from a water sample. And the best database is one built from the environment you're sampling. Ideally, I would have a reference database of all things possibly found at Mammoth Cave. I don't have that. And that's often not the case. But really, if you want to do long-term environmental monitoring of a particular ecosystem, you really want to put in the legwork and generate your own reference database. Go out there, collect specimens, generate barcodes from the actual specimens. That would be the ideal reference database. So back to the shrimp. These are the sites that we collected, 11 sites. Green are places where we detected shrimp. Red is where we did not detect it. And then a few of these black dots on here are places where our quality control failed. So these were sites where we might have decontaminated equipment in the field and gone out the next day. And we run blanks through all the sampling equipment, and we got positive hits on some of the equipment blanks. So basically we threw those out because we could not be assured that those positive hits were not due to contamination. So we found shrimp. We also found some other things. A few sites we found the Mammoth Cave crayfish here, Orconectes pellucidus. This is a random worm that I Google image search. There are a variety of other things in these data sets. You know, maybe these things are meaningful to someone. We don't really have the sampling to sort of come to any sort of ecological conclusions or habitat, species associations with the shrimp. We'd really need a lot more sampling. And then there's a lot of things that we can't identify in the data set. We actually can't identify more of the stuff in the data set is unidentified than stuff that it is identified. Other possible detections, this long list of things, are all sponges that are indistinguishable at the CO1 marker. And so some markers have limitations that they do not provide good resolution. Other possible detections, we found some green frogs. Cows is a common contaminant in these. And we might have also found ourselves in this data set. So at least this one data set, we found 13.9% of the identified rates, not counting the non-identified ones, were human mitochondrial DNA. Now these could have been ours, they could have been something from within the watershed. We learned some hard lessons moving in my lab from sort of taxonomy DNA barcoding to next-gen sequencing, that you always want to use filtered tips, particularly when dealing with amplicons, as we found a whole lot of freshwater mussel DNA, actually 2 different species that were part of an unrelated barcoding project. So at least in this particular sample, 95.9% of the identified reads turned out to be mussels. Now there were actually a whole lot more reads that were unidentified. So you might also get primer bias or potentially bycatch. So this one particular sample came back 80% chironomid midge. This could be that some insects actually got trapped on the water filter itself, maybe eggs or early instars, things like that. Or it could be that the primers really like chironomids. And so we have that primer bias in here. So again, back to the shrimp. So I mentioned initially when we started this, we didn't have reference sequences, and this project would have been ideal for a qPCR based project rather than a metabarcoding project. But 1, I didn't have access to a qPCR machine, and 2, we didn't have shrimp tissues to start out with. So when you have a master's student who's trying to get done, we couldn't really wait around for shrimp legs to show up. So we decided just to do this broad metabarcoding project and hope that we got shrimp along with in that data set and were able to identify it. Unfortunately, we were. Eventually, later on in this project, we did get those 2 little legs. And when you blast the results from that Sanger sequencing, this is what we get. Basically, there is a shrimp leg thing in here, but then all the rest of these are random arthropods, mostly insects. You see the percent identity is relatively low. So these are really just random hits. And so BLAST doesn't really work well if you don't have something closely related to it or the exact same thing in the database. So some people will use these hits and try to assign at higher taxonomic levels. I'm really wary about that. And this is one example. If I could not, I basically might be able to assign this to arthropod, but that's it. And so you have to be careful when things are not in your reference database. Back to our positive sites, we can do a little bit more with that. This paper was the first one to look at haplotype diversity of whale sharks. It actually came out the exact same time we were working on this workflow. So once you boil down the data and get rid of sequencing errors and things like that, and you're pretty confident that what you're seeing is real, you can look at haplotype diversity. And so we find these haplotypes lettered A through G. Most of them are really close related. Most of them are only 1 base pair different. And with the exception of this one G here, they all, I think, all translate identically. So the actual amino acid sequence is identical. There doesn't seem to be any geographic structuring with these haplotypes. So not sure how much that tells us right now, but we were able to do it, which I think is amazing that you can take a water sample and figure out haplotype diversity of this animal I've never actually seen before. But we don't really see any geographic structuring of those haplotypes. We have 2 sites, so we have negative detections. So it could be if they're extirpated from there, or maybe they're just in low biomass, or maybe discharge was either too high or too low that time for the DNA to get to us to where we were sampling. So this review is just type 1 and type 2 errors, so false negatives and false positives. I think for some of these, for causes of some of these, I'm particularly interested in karst environments in the ecological cause for false negatives. So we really don't know much about the ecology of the environmental DNA in karst systems. Does it last longer? Does it decompose faster? Do we have to wait particular times of the year or flow regimes to actually catch that DNA? We just, this is anecdotal, but we just basically plotted gauge height, so basically river discharge, and precipitation on different sampling dates. And so red is a day that we didn't detect shrimp when we thought we should have. Green is a day that we did. And so I was thinking maybe these pulses here would flush some of the environmental DNA from these organisms to closer to where we're sampling. Again, it's anecdotal. We don't know. It'd be great to do follow-up studies to look at the influence of discharge or precipitation on detection of karst organisms. Traditionally in riverine systems, we say don't sample after high rainfall. Might be the opposite in cave organisms. So I'm running out of time here. We need reference sequences, reference sequences, reference sequences. Any way that you can get them. If you're interested in doing this in karst systems, I would encourage you to do barcode blitzes and identify as many of the organisms from your systems as you can. That's really where the pressing need is. We can get DNA from a water sample, but identifying it is the hard thing, and the only way that you're going to be able to do that is if you have the reference sequences. I mentioned again the ecology of eDNA, particularly in karst systems. Some other things we're working on here. I don't really have time to get into them, but feel free to email me if you have any interests. And so that I can take any questions live from my house. So thank you.